Good morning, ladies and gentlemen. I am Kim Kyung-min from Korea Radio Stop Center for Pharmaceuticals. And everyone is getting into the room, so why don't we kick off this session, even though you are getting into the room because we are on time. And we have presenters here, so on the right side you can see me. And this session will take 55 minutes. And we have two presenters here, and actually the center director of this center will deliver his remarks. I'm a first presenter, and second presenter will be Lee Hyung-gi, professor. And also we are aiming to end the session by 1:30. So why don't we listen to opening remarks from the director of the center. Good morning and good afternoon, ladies and gentlemen. And I am Im Sang-bom, director of Korea Institute of Radical and Medical Sciences. Once again, my special thanks go to all of you for being here today. And actually, biodiesel global industry has been nurtured as one of the Korean government's industry, and it's put emphasis on the importance of this industry. And we are presenting seminar under the theme of new radioisotope technology. And we utilize radioisotope to support the development of new pharmaceuticals, thereby reducing dependency on global pharma, as well as fast-track the development of new pharmaceuticals. This is our establishment goal. And also we are aiming to supporting manufacturing of GMP and evaluating toxicity, and also we provide early support for early clinical trial utilizing radioisotope. And also we evaluate radioisotope in the preclinical trial, and also this is the one and only Korean government agency, and it's from life cycle support. We are utilizing radioisotope in the early clinical trial, and it's very honor for me to share our experiences with you. And at the same time, we have established promotion booth on the first floor, so you are always welcomed. So once again, I'd like to express my sincere gratitude to all the participants and presenters for being here today. And also, I'm looking forward to high interest in our center, and we are committed ourselves to providing support here. Thank you very much. Thank you for your warm welcoming remarks. And actually, I would like to introduce myself again. And actually, Im Sang-mo, our director. And I would like to begin my presentation. So, first and foremost, I am Kim Kyung-min from Korea Radioisotope Center for Pharmaceuticals. And for my first presentation, I would like to touch upon the technologies, especially radioisotope-based platform technology for accelerating new drug development. And our Korea Radioisotope Center for Pharmaceuticals, in short, KRICP, is our institute name. So this is a institution to provide radioisotope platform under the umbrella of Minister of Science and ICT. And also building construction complete in 2019, and we are operating our institution in 2020. It's been 2 years, so we have a lot of technologies for the drug development, but of which actually non-clinical trial and the early clinical trial are the areas of our expansion, and of which radioisotope technology is our focus. So we are supporting evaluation of safety and efficacy, and also radioisotope medical products are emerging as a new breakthrough. And also, we try to carry out tests for IND file. And we are a public organization, so we are very open to be operated. And also, we provide building facilities and equipment. This is our areas of support. And if there is a necessary for new technology, we are co-researching this, and we also provide supports if necessary. And as you're all aware of, we utilize isotope-based evaluation of pharmacokinetic and efficiency, and also safe toxicity and GMP manufacturing and early clinical trial. And as I mentioned before, we provide commercial services for researchers and companies, and among them, we provide necessary consulting as well. So radioisotope technology is something that I'd like to address, and radioisotope is consisting our human body. It is unstable, so it releases radiation. And so it is well used, long been used in the medical sector, and especially we try to utilize radioisotope that is very suitable and available in our eye. And also, this is capable of precise quantification of the amount of RI in test subject, which will be contributing to the development of new drugs. And also, there are long-held technologies for RI, so it allows for precision. And also, it becomes microdosing as well. So if it is emitted to our body, it is not detrimental. So actually, non-clinical trial in this setting, many people are worried about the dosing. However, you don't need to do that because we provide microdosing for RI administration, so you don't need to consider PK. However, you have to think about the characteristics of RIs, so this might be utilized in non-clinical trial supports. So especially safety, PK are the gold standard for our ID. And in bio, actually many people are suffering from difficulties with existing technologies. But if you utilize RI, you can solve it effectively and efficiently. And also, as I mentioned after, we have a video, so through graph, rather than imagination, we can see and understand the test. And also, this image can be provided in vivo and ex vivo, so this increases efficacy. And our center is providing all cycle support. So from the new drug candidate discovery to the IND, this is our focus. And we're preparing for phase 1 clinical trial, phase 1 clinical trial. We would like to expand our foothold here. So in the near future, there might be a RI-based clinical trial phase 1, an early phase 1 stage. So I will touch upon the activities that we are doing in non-clinical trial setting. So animal model, or we also evaluate the appropriateness and toxicity and efficacy and safety of drug candidates. This is our main focus, which requires us to ADM PK. And also efficacy and image is also playing an important role, image-based efficacy and image. And it is all applicable to other radioisotope pharmaceuticals. And also we use IRI marker or labeling. And we are the one and only to provide these services. And RI and bioimaging are effective. So actually, as you can see from the left top side, and this is conventional way, so we can evaluate ADM, but compared to the existing one, we can quantify the drug dose, a level, and which only be made possible by RI technology. And as you can see from many papers, you can see evaluation of RI distribution with time in each organ. and it's well known and it has well-structured data. But in preclinical trial, we can deliver this to the body. And actually, there is a challenges from animal model, but we can reduce mass by bioprinting technology, and also RI is the most accurate and gold standard for this. I talked about preclinical trial or non-clinical trial, and this should be effective and efficient and is very necessary with utilization of RI technology. Plus, we are pursuing, and other global leading companies are pursuing as well. So RI-based early clinical trials. So with the existing methodologies, you can see the focus with the razor point. But many articles say that clinical 1 and clinical 2, and some clinical trial 1 and 2 are successful or withdrawn. This leads to higher cost. But if you utilize RI technology, of course, this can be effective in preclinical trial, but there is a microdosing. So you can emit human body and animal model with our radioisotope. So it can reduce and eliminate uncertainties. And also, RI-labeled drug administration at a microdosing level, which is not detrimental to the human body, and it is very effective and efficient. And when you go into the phase 2 clinical trial, you can know something that is not known to us. So microdosing plus microtracing are utilized. And in combination with 2 clinical trial, it can take less effort and time to reach end goal in clinical trial. And this is the future direction of our research institute. And this will be delivered in more detailed manner by the next presenter. And KRICP, it's been 2 years since its launch, and we are in the third year. We have taken incremental approaches to achieve our goals. So microdosing-based evaluation on PK, and also we provide support in clinical trials. And we also provide evaluation of GLP toxicity to support clinical trials. And recently, actually, we provide our technologies to test vaccine in terms of hypersensitivity. And imaging-based evaluation is backed by our RA labeling technologies. Building on this, we also have partnership with businesses, thereby addressing their challenges. And also, we have supplied GMP-gradable radiopharmaceuticals for clinical trials. And as I mentioned before, when it comes to open lab operation, companies are beneficiaries of professional training services. And then I would like to take a deep dive into the RI pharmacokinetics and efficacy evaluation. So what's our activity here? So pharmacovigilance and PK and efficacy will be my topic. So PK, ADM, and PK are all utilizing RI. So our main focus is mass balance, and metabolite identification, and metabolite profiling, and PK analysis. And there are human and non-human, all covered by us. And also there is a structural change. So if you do clinical trial, you need approval. So GCLP is in progress to gain approval. And our center has been characterized by pharmacokinetic analysis of C14 labeled drug using AMS. But we also label C14 drug to analyze pharmacokinetics. 10 years ago, there has been a movement to apply pharmacokinetic analysis of C14 labeled drug using AMS in this analysis. And we introduced this technology. So for all drugs, if there's a carbon in drugs, it can be applied. So it can extract from preclinical and clinical trials. And not only PK and AEMS, we also analyze safety. it can allow you to evaluate safety in clinical 1 and clinical 2, phase 1 and 2. And also it allows for evaluation of metabolic inhibition of new drug candidate. So for all range of drugs will be applicable items by our technology. And not only copper and protein, but also other isotope can be labeled to evaluate ADME. So we can see biodistribution, and this requires no imaging here. And excretion as well. This is very important. So we provide excretion test of new drug candidate. For a non-clinical trial, we can monitor the absorption and the excretion of drug candidate in the animal body. And also, as I mentioned before, our center provides responsibility of bioimaging and services of bioimaging. And we have a lot of imaging modalities here. So we have been equipped with all things that you need. So it is all diseases are applicable for our bioimaging. And PK data analysis is one of our service. And in terms of pharmacokinetics, we set one mouse model, and we can see statistically meaningful data, and you can understand better with our imaging technology. And we can also apply this technology to large animal, up to dogs. And also in terms of disease model, there might be a model-appropriate test. And also we provide pharmacokinetic analysis, and per model, what kind of solutions can be provided. And this, we can also provide customized services. And we also create our disease model. And not only model evaluation, but also we provide radiopharmaceuticals dopamine here. So we also evaluate the development of therapeutics here. And not only that, we have a Padimari. This is equipment that is applicable to diseases that once thought to be unapplicable by existing or legacy equipment. So with this Padimari, what is interesting is that you can see the imaging and biodistribution more vividly. So let me wrap up my presentation. We are developing ways to make most of our eye technology. So this can be all providable to everywhere in Korea. And we are in partnership with relevant agencies, and we are doing our best to provide service, and we also have been equipped with proper networks. So if you have something that you need from us, then please feel free to reach out to me. Then we will provide our consultation and support. So if you do have a question, so please contact Korea Radioisotope Center for Pharmaceuticals. And you can see the tab, and you can see the homepage, and you can see the overview of our institute, and also infrastructure, equipment utilization, and test requests can be seen. So please feel free to reach out to us. And if necessary and possible, we will do everything we can do to support you. And you can see the brochure on your table. And actually, the brochure is not telling everything about us, but please see your brochure and please reach out to us. And next, Professor Lee will cover the presentation.
Professor Lee is a well-known person, and he is a professor at Seoul National University Hospital, and he is doing a lot of jobs. And from the U.S. to Korea, he participated in lots of clinical studies. And using carbon-14, he is one of the pioneers in Korea. So he is collaborating with us and provide lots of recommendations. So if you have any questions, then Professor Lee will give the answer. Can you listen to me? Thank you very much. Thank you very much for the warm welcome, and thank you for the invitation, Professor Lim, and thank you for the researchers in the center. This is the first page. As I introduced, let me introduce myself once again. I've been participating in the clinical studies, and I was also part of the FDA for a year. When I was an associate professor, I was a fellow researcher, and I wrote lots of books related to the new drug development in Korea. So how we can achieve the K new drug development. And there are some English books as well. And I believe you are familiar with this slide. It takes a long time to develop the drugs, and there are lots of risks. And in each stage, there are many risks and reason for failure. Of course, when we conduct clinical studies, the first problem is safety, but eventually we are faced with the efficacy issue. So in the preclinical and non-clinical study, and then it is moved on to the first phase. So here, PD and PK are the main problems, and that is the focus area for me. So it is very obvious, and many people understand this. However, still, many people do not focus on this. In the early stage of the clinical study, we need to focus on the PD, PK, and there are many metabolites. So we need to understand them. And the relative mass of the metabolites is important. As you know, PK and PD are well known. When it comes to the pharmacokinetics, it is about what our bodies does to the drug. But when it comes to the PD, pharmacodynamics, it is about the handling of the drugs to the body. So if it is well handled, then it is well observed. But if it is not the case, then it is about the toxicity. So handling the drug inside of the body will take 4 stages: absorption, distribution, metabolism, and excretion. So when it comes to the absorption, we measure this by plasma compartment. And when it comes to the distribution, it is going through a whole body. And the metabolism is including drugs, xenovirus. In order to excrete it outside of the body, the developed process is evolving. So in order to excrete it rapidly, we need to dilute it into the urine. So a dissolution in the water means it should be charged. So most of the process in the metabolism is making it charged. And whether it is charged or not, it should be excreted through the bowel movement or urine, or sometimes it is expressed as a bump. So we take the medicine orally, and it goes through the whole body. And I simplify the process. But actually, it goes through lots of process. When it comes to the GI tract, many people think that this is part of the inside of the body, but actually, GI tract can be seen as an external part of the body. But anyways, this process is very complex. And as I mentioned before, there are 4 processes in the PK, and as for the PD, the drug inside of the body should show some effect. Depending on the concentration, there might be some actions of the drugs. So when we develop new drugs, eventually we need to have the PK dashboard. So in which process, how much will be injected into the human body, and how much will be metabolized, and how will be excreted. This is the whole process, and we also need to understand this. That's why PK dashboard is important. That is the dream of all the new drug developers. However, when we use the specific material with isotope, then we can effectively understand this. There are many things we can do. For example, we can do the PK study, and for me, in the early stage, we test a single dose and multiple dose, and when it is non-IV, then we can test it. But there are some limitations. First, we need to see the route of excretion and how much will be observed in the entire body. So it is called as absolute bioavailability. And what is important here is inside of the body, how much will be metabolized and what is the outcome of the metabolism. The authorities, including FDA, will see the tox data of the animal test. So if the toxicity is now observed in the animal model, or even if it is observed in the animal model, it is even more elevated in the human body, then there will be disproportionate metabolism. Then the regulatory body will say that this is different. So we once again need to confirm that toxicity is appropriate. But these are the limitations of the PK study. That's why we are using labeling. We use isotope. There are C14, and as for protein, sometimes we use a phosphate. But C14 is mainly used, and this is the half-life. It is easy to create, but the problem is when it is met with the oxygen in the body, it will turn into water. We use C14, but unlike the conventional way, I provide microdosing. So that's why I'm called this microdosing or microtracing. There is not that different. Cord compound will be given together with this isotope, then we can trace them. That's why I call this as microtracing. But they are almost a similar concept. So small dose will be given to human body. So 100 microgram will be given, or only 100th of the effective dose will be given. And with the labeling, we can obtain the PK-related information. Then why we need to use micro? Conventional method is already available. We can use the conventional method. Then we can use a microcule level. But we are using nanocule level, which is smaller than 1 microcule level. And when we give microcule level isotope, then before human body, we do the quantitative whole body autoradiography study for the animals. So C14 labeling will be given to animal, and in a time series manner, we freeze them, and on the plate we see where the isotopes are located, and the dosing material will be used. So we will tell whether it is okay to put into the human body, but it takes time. So these are conventional methods, but if we provide nanocurie and microtracing, then we don't have to go through this process because the radioactivity is very small, but the volume is really small. So liquidation counter, LAC, cannot be used. So AMS, a specific machine, should be used. And to easily put, we have a strong magnetic power here, and depending on the weight of the material, the light one will have a larger curve. C14 is given in a small volume. So in a sample, if C14 is contained in a large concentration, Then we'll compare this with the naturally located C14, and we will look at the plate and tell whether it is a C12, C13, or C14. So let me tell you why I'm interested in this area. Everything began here. I came back to Korea in 2012 for this project, and I contributed to this project to be commissioned to Seoul National University, and we created this Connect. And I also created the name of the center. And Seoul National University Medical Center received this project. And microdosing, microtracing was the main project. And I didn't realize that what is the main content of this project because I didn't study about this in the US. But it was a serendipity, so it was given to me. So I decided to go ahead, and I became the responsible person for this project. And other doctors did not have the interest in this project, so I was the last person who can take this job. So eventually, we took the project and changed the regulation, and we also changed the laws as well, and we also did the promotion. Actually, my students and our specialists are doing most of the job, but I participated in this photo shoot. And we have two studies, microdosing and microtracing studies. And for the first four studies, we conducted the study in an IIT manner, and the rest of the three studies have been conducted in a SIT manner. So when we talk about this study, people got interested in this project, and now sponsors are also interested in this. So they are knocking on the door of our hospital. And one of them is FDA IND opening study. Of course, we submitted the IND, and without hold, we received the reply. And when it comes to the IND, we need to submit Form 5072. We need to submit all the experiences. So it is because the FDA wants to check and confirm whether there is any criminal record in my experience. So I was getting the green light, and I initiated the project, and we established the system. So we established the SOP, and for the first time in Korea, I received the IND approval. So I'm the IND holder. So my name is located here. So I'm the first person, IND holder, for this microdosing study. And this is about a radiology safety report. So one corner of the radiology department was dedicated to C14, and we established the equipment, and I participated in the photo shoot, even though all the works have been done by our specialist. As Dr. Kim said, mass balance is about excretion of the drugs we gave. Of course, 100% collection is the ideal number, but it is not possible because some of them will be excreted through the respiratory system. So over time, we see how much will be recovered. Over time, the concentration of the drugs will be indicated, and using C14 or other radioisotope, we can see the total radioactivity, and we can measure this. This total radioactivity is the sum of the parent and certain kind of metabolites. So this is the combination of the parents and metabolites. So if we separate them, we can separate the parent from the metabolites. So the metabolites part can be the metabolite burden, and if it is too big, that is the problem. So from urine and bowel movement, we can see and we can collect the drugs recover, and through a radiochronogram, we can measure the concentration, and we use quantitative and quantitative measure. So inside of the peak, we can have the area, and this area shows the metabolites. And we can tell whether it is plasma or urine. This is the study we did, one of the new drugs for obesity. As you can see here, this is the treatment for obesity, and we gave only 0.6 microgram. So we use 200 microuri. For the microtracing, we gave a fifth of the original volume, but still we were able to detect it. And I also participated in the photo shot, and we also imported this special equipment to measure the excretion. This is the total amount, and it is almost at 90, even though it is not 100. So the study went well. And if it is excreted urine, we can see the bioavailability of this drug. So in order to have the urine, first it should be observed. So we can tell that this drug is well observed. I was the PI, and we need to see and separate the parent and the metabolites and compare the metabolite burden with the parent, and it is very small. Most of them are excreted through urine. So most of the urine is containing the metabolites. We wanted to have the ID, however, it was not possible. Technically, it is very difficult, so we did profiling, and through UV, we measured and compared this with the parent, and this is plasma versus urine. And when it comes to the urine, the parent showed a peak at the early stage. So this is mostly metabolites. And you can see the big gap between the parent and the plasma. So in this case, we cannot observe this in animal model. In this disproportionate metabolites, we can measure the area or the curve of the parent concentration. And before we move on to the next phase, we can tell whether it is toxic or not. So this is a metabolites in safety testing. So for this company, I recommend it to go ahead. But they ignored my recommendation. But anyways, in animal study, most of the drugs will be excreted through bile. However, in human, it is excreted through urine. So the accurate model should be human. When it comes to the rats, it has a higher bile excretor. And we realized this for the first time. And I would like to talk about the absolute bioavailability. When we injected with IV, the total amount will be 100, and how much will be available in the systemic circulation. Of course, we need to adjust the number, and the best way to do that is cross over these two ways. So I should be our own reference. So one time we inject with IV, and then we adjust the number. This idea is correct, but there are limitations. So I should be the same before 1 month and after 1 month. But it is impossible. But I can say that it is possible. Healthy volunteers are usually those in the 20s. So they are uncontrollable. So they sign on informed consent, but they do not read the fine print. So in the first phase, they participate actively, but in the second phase, they drink alcohol a lot, and their body condition is totally different after only 1 month. But they think that their condition is totally same. But there is a bigger problem. First, in order to do this, we need to have the IV formulation. When it comes to the IV formulation, the material should be soluble in liquid, but many materials are solid, and there are many problems in finding out the media. And in many cases, drugs are developed in an orally administered drugs. So we also need to ask them to turn it into IV. But we can address all these problems using micro-tracing. First, unlabeled oral drug will be given in a cold form. Then, the small volume of isotope will be given in an IV manner. Then, whether it is labeled or not, since it is same compound, they will circulate together. And we can do the micro-tracing, and we take the blood, and these blue ones are conventional mass, and micro-tracing C14s can be measured together. And obviously, the volume is really low, so we need to adjust the number. Then we can see the concentration profile depending on the time period for hot and cold, and we can also understand the absolute bioavailability. So we can put them in one chart, and even if it is not soluble, a small volume can be soluble in liquid, so there will not be a problem, and we don't have to do another toxicity testing. Let me give you an example of Korean company. The ulcer treatment was developed by this company, and from placebo to 800 milligram, there are many doses, and the company allowed us to give a green light, and phase 2 was already completed. We didn't realize that, but we did the study. So the company allowed us to use this example for my study. We gave 80 microgram of the IV infusion. And this chart shows the oral administration and IV administration. And the absolute BA is around 10%. So bioavailability is very important. 90% of the bioavailability can be the gap. So it is not observed, or sometimes it is observed twice. And we also did pharmacogenetic studies. And there are ups and downs in this graph because due to the physical properties, there are some crazy parts. And there are highly variable parts. So in the initial stage, we need to confirm the PK profile. the company allowed me to do the study, but already the company made a lot of investment. But I received 15 million Korean won from the government to do the study. And in our field, CPT is a highly reputable journal, and I published this. And it is almost new to the word biologics. Hydro FC portional IR7 has been fusioned together as a protein, and it works. So IR7 is attached together, and it proliferates the T cell in our body. And I was the PI in the first animal study, and the first component is calculating the absolute bioavailability, and the second one is calculating the route of excretion. So I conducted the study, and we can see the hot line and cold line. And this is before the adjustment of the number. Approximately bioavailability is only 2% approximately. So it seems that it is not bioavailable. However, we also need to see the excretion from the body. 85% are coming through the urine. So you may have a question. Now, this is given as a subcutaneous manner, and exclusive pathway is not the route it took. Lymphoid organ or lymph node or spleen was used, and eventually it is going out of the body. So we can use the isotope for this case, and we can also see the fate of this material. So in the future, there will be more drugs. The red area is showing the drugs that is using AMS. In other countries, they can do the same thing, but in Korea, it is much more cheaper. And in the future, this will become more important. And I'm grateful that including Professor Lim, our center allowed me to do the study by establishing the one-stop center. And many people provided the support, and I'm just holding the banner. So the industry and academia provided the support. And we have Mr. O here. We signed an MOU with BioCoa, and we are carrying out studies together, and BioCoa is doing the measuring. For this, we participated 4 international symposiums, but we don't have any more projects now, so we are not holding the symposium anymore. But I hope that the center can continue this work. And so many people provided the support, so I wasn't able to include everyone here. But if I knew that this many people should participate in the project, I would not take it. For some projects, it takes 3 years, and only 1 day before the 3 years, I received the IND. So sometimes I take money out of my own pocket to conduct clinical study. But anyways, there were many difficulties, but I'm happy that we carried out a microtracing study in Korea, and we also plan to do that. And companies are giving attention to this. And what is more important is the Food and Drug Safety Administration is recognizing that this technology is very important. So it is providing support in many ways. And also, the center is providing technology, equipment, building, and human resources. So I'm not doing this alone, but I was able to contribute to this. This ends my presentation. Thank you very much.
Thank you, Professor Lee, for your detailed and thought-provoking presentation. It was very easy to understand. And our luncheon session needs to be ended at 13:20. So actually, we are able to shorten our time. So if you do have any question for presentations of Professor Lee or for our center, if you do have any question. And actually, we already carried out first and foremost study, and we are able to confirm final dosing. So what I would like to suggest is that many companies are turning to this direction. So first, animal study during this study or extended as well as part of extended cohort, we are suggesting this. And microtracing requires cord form dosing, and that dosing basically is one injection. So single ascending dose from single ascending dose in phase 1 clinical trial, we have to secure safety tolerability, but we have to lower the dosing that is very lower than upper margin. And so this is depending on the purity and non-radiation nature. So we can provide microdosing if it's featuring higher purity. So as I mentioned before, therapeutic dose is, let's say, more than 10 millimole, or some up to the 100. But our microdosing, by definition, is 0.something milligram. So our microdosing is less than 0.1 milligram. So we make it desorbable and we make it diluted. And at the same time, as I mentioned before, in terms of the radiation, so AMS is very highly sensitive. So something that I mentioned, among them, 80 microgram. In case of this, 1 microcube is like this. However, when 200 nanocurie can be admitted, but it can also suitable to detect in our body. So it is very highly sensitive methodology, and it is with a high accuracy. So by definition, that's right. But in the first animal study, as a one arm or as an extended cohort, if we utilize microdosing in this, we utilize this to cohort that secures of dosing tolerability, and it can be applicable to the minimum dose when we secure tolerability. If you do have any question.
Yes, please go ahead. Professor Lee, Professor Kim, thank you very much for the presentation, and it was very important topics. So I have many questions, but I would like to ask an urgent question. Using RI in the clinical and non-clinical study can be compared with other studies, and in terms of cost or in terms of time, there might be differences. And those who have experiences in new drug development, they may ask that why do we need to go through this process? And for those opinions, how we can convince them so that they can go through these IR-related studies? I'd like to ask your opinion. CT is one of the important components. So whether we will use a highly radioactive material or low radioactivity, and ABA in the past for the non-IV, it was not mandatory, but now it is included in the package, and many countries are following the suit. So we cannot say that it is not necessary. And when it comes to the cost, C14 labeling is very important. Anyways, in the preclinical study, we need to have a C14. And when we inject it into human body, there should be repurification to enhance the purity. And it does not take additional cost. And as for a mass balance study or ABL study, we use only one cohort, and usually it consists of 4 to 6 subjects. So the cost is not that high. Of course, in the past, analysis took a large cost, but for the concentration analysis, it takes 4 cents for one sample. So it was about $200, but now cost is lowered to $100. So cost is not the problem nowadays. And increasing number of the regulators trying to use the information that can be only obtained by tracing, identification of the metabolites and profiling of the metabolites are required, and we need to identify the structure. So the peak of the radiogram is absolutely needed. So increasingly in this field, regulators show their interest and they will demand more. So for those who say that this is not needed, you are outdated, and in the future we will need more.
Thank you. Actually, Professor Lee elaborately mentioned this. Actually, this is very sharing the same concept in the preclinical trial, and plus I would like to mention affordability and cost efficiency. And utilizing RS costs a lot. However, actually, when we estimate the cost, we have to consider time and effort into consideration, and we can cut 1/5 of that, including drugs. So we can also reduce the amount of those drugs and time and efforts as well. So if this becomes a reality, we can reduce 1/5 of the cost. And technologically, as previously mentioned, biopharmaceuticals. In terms of this, ADME is an area that is not solvable by existing approaches. So at a national level, there is a high expectation on biopharmaceuticals, and also if there is a plan that we have to utilize RI technology, this might be partially or completely be a part of this program and agenda. And we have 2 minutes left, so.
My name is Song In-ho from Bundang Seoul National University Medical Center, and I'm in the same field. So my question is overlapping to the previous question. The ABM, TPK, PDA can create the data, and companies ask a question. And they think that the difficulty is whether it is appropriate to submit the FDA and Food and Drug Safety Administration. When it comes to the sophisticated drugs, ADMA cannot assess the drugs perfectly, such as the exosome drugs and protein drugs. So can we apply this technology to those drugs? Professor Kim, first.
So for the sake of time, I will short mine. So of course, there's a mandatory requirement as well, but at home, actually, this is not the situations in Korea, but realistically, we have to establish backup data, and it costs a lot, and my experiences taught me. And developers should validate RI data after they confirming this is effective. So as you mentioned, exosome-related issues that are applicable. And I'm on the same chapter, and so domestic companies coming to me is not intending to submit to Korean MFDS, but to the US FDA. And one of the biggest issues in biopharmaceuticals is that bioanalytics are not able to be set up in a proper manner. So actually, the third example that I've shown to you is like that. So labeling allows us to accurately evaluate concentration. We have utilized many variety of ELISA, overlap antibody creation or detection, and indolence compounding. When let's just say this is question like R7 is very difficult. However, we can overcome these barriers, and PK profile and accurate analysis of PK profiling can be only done by RI. If you do have further questions, but we have a lot. Actually the time is done. So thank you once again for taking time out of your busy schedule. And if you do have a further question, please reach out to our booth to E24, the section where our booth is set up. So please feel free to reach out us. And if you do have a question, we will happy to answer your questions. And also, please come to visit our booth. And if you do participate in a survey, then we will provide a token of gratitude. So, and once again, I wish you enjoy this session later on. Thank you very much.